So when replication occurs, every effort is made in order to make sure that when we are copying the DNA, that we are maintaining the message. There's a lot of going back and checking and double checking and proofreading to make sure that we are putting in the right base pairs when we're making copies, because when there is a difference in the order of the sequence, it is called a mutation. And this mutation can result in not producing a protein that might be necessary for cellular function or for cellular viability. However, mutations sort of always get this bad rap that like any mutation is going to be deadly or bad, but mutations are actually what is the driving force behind evolution. So mistakenly putting in a wrong base pair could also create something that is advantageous or give an organism some trait that allows them to survive better in their environment. So, an organism that is in its natural sort of state is called a wild type organism. So it's a non mutated organism. An organism that has some sort of genetic difference is called a mutant strain, and it has some sort of like mutation. And a lot of times these are really useful for us to sort of look at genetic markers and to sort of pinpoint what genes do. So if we had a wild type organism and then we had a mutant strain, whatever gene we had messed up or eliminated or whatever from the mutant strain, we could see the difference between the phenotypes of the two organisms, and then infer something about what that gene what its job is. Now, we do have ways within the lab where we can actually screen for uh, for mutations. So by using a selective medium, we can actually select for different mutants. So the different colors here just demonstrate a different kind of media, and we're growing the bacteria on the media. We can expose it to some sort of mutagen or something that's going to mutate or change the DNA, and then we can select for the mutant strains by growing them on a plate that will only allow the mutants to grow. Whereas over here, you would have a non-selective media, which would have a mixture of wild type and mutant strains. So just to kind of point out another example of using selective media in order to separate out different kinds of organisms. Right. So mutations can be spontaneous, meaning that it's just a mistake in replication, putting in the wrong base pair and not fixing it, or eliminating or adding in an extra base pair and not fixing it. Or we can induce mutations by exposing organisms to mutagens. It could be anything from like some sort of radiation or a chemical, something that can get in and alter the DNA. So a point mutation is when we have either added, deleted, or substituted in a wrong base at a single base, right? So the addition and or deletion are really bad typically for the organism and cause what is called a frame shift. But we can also have the substitution of a single base, which can lead to several different things. By substituting in, let's say, instead of putting a T, you put an A, right? This change could cause there to be a non-functional protein. So this is a missense mutation, which leads to a different amino acid going into the sequence. Now, sometimes it can cause very little difference in the function of the protein. Sometimes it can allow it to function in a different manner, which could be beneficial, or it could create a non-functional protein. So this is a missense mutation where you put in the wrong amino acid. A nonsense mutation is one in which you have changed from a normal codon to then being one of those three stop codons. A silent mutation is one where you change the base pair, but it doesn't affect the amino acid whatsoever. So remember, if we go back, let me go way back up here and see if I can find that table. Here it is. So let's say if in the third position we put a C instead of a U here, we'd still code for leucine, and so, it would be silent. We wouldn't see any difference. Here, remember, these are our stop codons. So if maybe you were supposed to have UAC, but you changed it to UAA, then that would be where we'd have nonsense, because we'd go from coding from amino acid and continuing to completely cutting off the entire sequence. All right, so let's go back then and now talk about frame shift mutations. So frame shift mutations is when you actually delete or add in an extra base pair. And what this does is it throws off the entire reading frame, so every single one of your triplets is messed up, and it almost always results in a non-functional protein. So if your wild type was this set of triplets, to just sort of give you an idea, you've got, the big bad dog ate the fat red cat. Well, if we had some sort of substitution mutation, maybe we changed this A and we had an I instead. So we have a missense where we've made a different protein or a different phrase. The big bad dog ate the fit red cat. But, it changes the meaning a little bit. Maybe it could be better, maybe it could be worse, right? Um, and then you have a nonsense where it stops. All of a sudden, the big bad, you cut off the rest of your amino acid sequence. This is pretty bad. Now, frame shift mutation, if we add in just one letter, see how it messes up the entire frame. So instead of saying the big bad, now we've inserted a B here, and so it throws off every other triplet codon. Whereas if we deleted, the same thing would happen. The A has been deleted, it throws off every triplet, and it doesn't make any sense at all. So frame shift mutations by insertion and deletion are pretty detrimental to the organism. All right, so there's a way in which we can screen for different compounds to determine whether or not they're considered mutagenic. And typically when things can mutate DNA, they can also be considered carcinogenic. Now, bacteria are the DNA structure of bacteria is very similar to the DNA structure of humans, right? So of eukaryotic organisms. So we can perform what's called an Ames test by exposing bacteria to chemicals to see if it mutates or is a carcinogen, and then assume that that would also mutate mammalian or like human DNA. So the Ames test is a way of screening different chemicals to determine whether it would mutate DNA and be potentially hazardous to us. Now, mutations are permanent, right, and inheritable. So if they end up in the DNA, then they're going to pass to the offspring. Most mutations are harmful, but some allow for you to have an adaptive advantage. Like maybe you create a mutation that makes you antibiotic resistant. Then that's something that allows you to survive better, and then you can pass on to, future generations. Now, there is the possibility of us controlling mutations, right? So inserting or removing or changing the gene expression of some organisms. And this is called recombination, when we donate DNA from one organism to another and causing another new strain. It could be through bacterial plasmids or it could be through gene exchange, right? And this organism that now has this new bit of genetic information is called a recombinant organism. Usually it's small little pieces of DNA, chromosomal fragments, or plasmids, and it can happen by either conjugation, so we know that's using a pili in order to move a plasmid from one organism to another, or by the process of transformation, where you basically just expose or upset a bacteria a little bit and their membrane becomes a little holy, and it allows the DNA actually to move in through the cell wall or in through the cellular membrane. And then a specific piece of genetic information that we want would go into that organism. Now, a lot of times this is done with either bacteriophages or, which we talked about before when we talked about viruses, but it also can be done by using plasmids. right? Now, this can be negative in terms of bacteria commuting antibiotic resistance to one another, so it's negative in terms for us, but we can also force organisms to express genes that would be helpful for us, like we can have bacteria express insulin genes so they can make human insulin, right? So this is the idea of like genetically modified organisms, but keep in mind that it's not all bad, that being able to control the gene expression of an organism that we can control in the lab or could benefit people isn't always a negative thing. All right, so that's the last video on genetics.